morning, dear students. Today we are going to see the effect of the gradient of dorsal protein in the development of Drosophila. So we are going to consider the embryo of Drosophila just finished 14th cycle of division. As it finished this 40 the cycle of division, we know that all the cells, there's enough number of cells, and it will become a blast stage. Certainly will become a blast stage. And if we take a cross section, it looks like this way. And for explaining all the process, what is the effect of the gradient of dorsal protein, I am drawing that cross section of that embryo in this form. So in this form, it will be easy for me to explain all the changes. So the cells with more amount of dorsal protein, not dorsal means words, more amount of dorsal protein. That cells will become mesodome cells. Which cells will become mesodome cells? Cells receive maximum more amount of dorsal protein will become the mesodome cells, and that cells will be positioned in the ventralmost region. So the cells in the ventralmost region, they receive more amount of dorsal protein, and they will become the mesodome cell in the further development of the Drosophila. Clear? Then, as we examine the cells up from here, these cells receive only less amount of dorsal protein, and they will transform to glial or ectodermal cells in the further development pathway of the Drosophila. So, I'm going to explain you about the first morphogenetic movement, that is the invagination of that ventralmost cells to form the mesodome. So, I'm going to explain how that invagination happens, everything like that. 16 ventralmost cells. It's not possible for me to draw and show all the 16 cells here. Anyway, you need to understand that 16 ventralmost cells, they invaginate because of some process, because of some changes happened in the embryo. They can invaginate and form, and from that invagination later will develop body muscles, fat bodies, and gonads. Clear? Then how that invagination, how the gradient of the dorsal protein result helped in the invagination of these ventralmost cells. So that's what we are going to see. So the dorsal specifies these 16 cells to form the mesodome. Again, that schematic representation. For your better understanding, I just drawn the cells in all regions like this way. So here you can see the dorsal protein that formed from the dorsal gene. This dorsal protein have 5 target genes. It can activate 5 target genes. Which are those genes? You need to note those names of all the 5 different type of genes. They are PIST, snail, FGF8 receptor gene, then rhomboid gene. So dorsal can activate all these 5 different types of genes. Okay. Among these 5 genes, the transcription of this twist and snail, it is very necessary for the development of mesodome phenotype and proper gastrulation. So these 2 genes, its transcription is very crucial in the development of mesodome. To transform that ventralmost 16 cells to mesodome, transcription of these 2 genes are crucial. So I already told you that dorsal can activate all the 5 genes. Okay. So why these two genes are very significant? How they play an important role in the transformation of 16 ventralmost cells to mesodome. So how the twist gene transcribed to give rise to twist protein, and that twist protein activates all the mesodermal genes in the ventralmost 16 ventralmost cells. Clear? So, in what way it support? It helps the 16 ventralmost cells to become mesodome by activating the mesodermal genes that present in the 16 ventralmost cells. They help the help those cells to become mesodome. At the same time, what the snail gene is doing here, the snail gene, as it undergoes transcription from here, a snail protein will form, and that snail protein repress is not activating, it's repress what? Non-mesodermal genes in these cells, in the 16 ventralmost cells. It repress the expression of non-mesodermal genes. By repressing the expression of non-mesodermal cells, mesodermal genes, indirectly it can support the development of mesodome from the 16 ventralmost cells. Then which are those non-mesodermal genes? This FGF8 and rhomboid are non-mesodermal genes. So these two genes are repressed in these 16 ventralmost cells. Clear? So I told you in the beginning that dorsal can activate all these genes. That means in the initial phase, these genes can also get activated in these cells. But as the distant snail protein is transcribed, this snail protein repress the expression of these two one and prevent its expression, thereby supports the development of mesodome from this 16 ventralmost cells. So that is one way how the dorsal help in the transformation of 16 ventralmost cells to the mesodome. Is what mentioned here. So rhomboid and FGF8 are not expressing in the ventralmost cells, but it express. But these two, rhomboid and FGA, will express in the cells adjacent to that place, adjacent to that 16 ventralmost cells. In those cells, these two gene will express, and their protein will form there. And as their protein form in these cells, what will form out of these cells? What means what type of cell will be formed out of these cells that is positioned here? From here, presumptive neural ectodome will form from the cell seen up, just up from the 16 ventralmost cells. Okay. Next, I'm going to tell you about the nucleus of the ventralmost cells. Nucleus of the ventralmost cells. So we know that the ventralmost cells means those cells that we can see somewhat here. And I'm going to talk about the nucleus, about these ventralmost cells which receive high amount of the dorsal protein. So as these cells receive high amount of dorsal protein, and these cells also will have high amount of twist and snail protein, because this dorsal activate these genes to form these proteins, to stand snail protein. So by that way, certainly in these cells, its nucleus, the concentration of twist and snail will be high. Okay. So that is what is mentioned here, that this concentration of the twist protein is high in the nuclei of ventralmost cells. And because of that high concentration, it can activate FGF8 receptor gene. Because of the high concentration of twist, high concentration of twist, In these genes, which gene get activated? The FGF receptor, FGF receptor gene. FGF receptor gene got activated and it express and form the FGF receptors in these cells. So, FGF receptors will form in these cells, 16 ventral-most cells. Okay, how these receptors are formed? Because of the high concentration of this protein, it activates the FGF receptor gene in the 16 ventral-most cells, and because of that activation, that gene express, it undergoes transcription and form the FGF receptor in these ventral-most cells. Clear? And in the cells, just see near, means up to this 16 ventral-most cells. In those cells, the concentration of dorsal protein will be less when compared with that of the 16 ventral-most cell. So, by that way, in these cells, it is possible for the FGF8 gene to express. So, FGF8 will express in these cells, that's seen just above that 16 ventral-most cells. And this FGF8 protein formed from that FGF8 gene, it is received by the receptor. I have just shown only one receptor now. This FGF8 protein is received by the FGF8 receptor that present in the ventral-most cells. And after the reception of that FGF8 protein or its binding, then this 16 ventral-most cells start to invaginate. So that causes that 16 ventral-most cells to invaginate. What causes? First, dorsal protein. Ventral-most cells first it receive high dorsal protein. Because of that, it get a high concentration of TIST, and this TIST results in the activation of FGF8 receptor, and in the nearby cell FGF8 protein is formed. It is received, it bind to that receptor, and after that binding, that 16 ventral-most cell in vaginae. That's the first morphogenetic movement. How it happens, I think it's now clear to you. Okay. Next, I am going to tell you about those cells that is seen in the lateral side, and these cells are lateral to the ventral-most 16 cells. So, what happens here? These cells receive only intermediate level of dorsal protein, and this intermediate level of dorsal protein results in the activation of another gene. What is the name of that gene? SOG. So, SOG gene got activated in these cells, and these cells will transform to neural ectodermal cells. So, after it become neural ectodermal cells, followed by the invagination of the 16 ventral-most cells inward, these neural ectoderm cells can move in this direction and cover the bottom portion of the embryo. Clear? So, first that invagination happens, and later then in these cells, because of intermediate level of dorsal protein, SOG gene activated, and because of the formation of SOG protein, these cells start to transform to new ectoderm cells, and they start to move and cover the bottom portion of the embryo. So, that is mentioned here. So, protein prevents the ectoderm in this region from becoming hypodermis and begins the process of neural differentiation. After the mesoderm cells invaginate, these neural ectoderm cells, they positioned at the bottom of the embryo. They positioned at the bottom of the embryo. Okay, clear? Next, I'm going to talk about another mode of action of this dorsal protein. So far, I told you that dorsal protein have an activating effect over 5 target genes, and I mentioned the names of those genes in the beginning of this section. While stand as an activating protein. It acts as an inactivating protein also. It has an inactivating action also over some genes. Which type of genes? Dorsalizing genes. So in that ventralmost cells, this dorsal protein at the same time it activates twist and snail. At the same time it repress certain other genes. Dorsalizing gene, dorsalizing genes, dorsalizing genes. So, which are those dorsalizing genes? I will explain to you. Dorsal, and this dorsal I can inactivate some dorsalizing genes like not these names Zen and DPP. So these are the dorsalizing genes. As these genes are suppressed in this region, these cells will not form any of the dorsal structures. From that statement itself, it is clear that here in these regions, cells in these regions, these genes will not get suppressed by the dorsal protein because these cells only receive a feeble small amount of dorsal protein. So that protein is not enough suppress that dorsalizing genes of this area, these cells in this portion, and that portion can develop to dorsal structures, dorsal part of the embryo. Okay. So thus in the same cells, the dorsal acts as activator genes as well as the repressor of some other genes. How the dorsal repress this dorsalizing genes? That is also we need to understand. Just shown you the schematic representation of Zen gene and in its enhancer region. Enhancer, I hope that you know what is enhancer. Enhancer is that portion of any gene that have a regulatory role. To that enhancer portion, transcription factors can come and bind, and because of that binding, it can regulate the rate of transcription of that gene. That is the role of enhancer. Anyway, leave that. That's not our topic. Look, in the enhancer portion of the Zen gene, there are some binding sites. These are silencing site. Its name is silencing site. Okay. In this site, first the dorsal come and bind. Followed by that binding some other protein also bind the in some other binding site that present in the enhancer, and because of this binding of some other proteins, the dorsal become able 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 to bind another protein. What's its name? Grucho. That is a as a repressor, as a protein that can repress the transcription of this Zen gene. So by binding with that Grucho, the dorsal can repress the expression of Zen. So those embryos that lack DPP and Zen proteins, they fail to form the dorsal structures. Okay, then I'm going to sum up all the points. In mesodome precursor cells, which all the genes will express. Now it's clear, very clear to you. Without my means brushing up, you can recall it. Which all the genes? Twist and snail. The twist and snail gene will express in the mesodome precursor cell. There is no Zen or DPP. Why? Because the dorsal is suppressing those genes and those genes expression there. Then in dorsal epidermis and aminoserosa precursor. In this portion, which genes which genes have the chance to get express Zen and DPP? There is no twist or snail because there is no sufficient level of dorsal protein. So that gradient is working to create all these different layers. Okay, and in glial precursor cells, here there's which all genes are expressing twist and rhomboid. Twist and rhomboid is expressing there. And in lateral ectoderm, we have seen that there is no expression of any of these four genes. Instead, what is expressing there? Zog. Zog gene is expressing, and it will prompt those cells in this position to form the neural ectoderm. So that is what about the effect of the gradient of the dorsal protein in the development of Drosophila. I hope it's very clear to you. Just going to wind up today's section. Thank you.
I'm an icon, man. I'm a girl about a 10. I should fuck her in a Benz.